Hello, welcome to Bite Size Med. This video is on the resting membrane potential in neurons. Couple of things before I start. One, any numbers I use are just to help me explain it. Numbers vary between books. Two, I'm going to use letters to represent the concentrations. So bigger, bolder letters means it's a higher concentration, and smaller, finer letters mean a lower concentration. Right, now I'm going to try and explain the resting membrane potential. Resting membrane potential. Let's remove the resting part of this and understand membrane potential first. It's the potential difference across a cell membrane between the inside and the outside. The membrane potential is on the inner side of the membrane compared to the outside. So the outside is used as a reference and is assumed to be zero. And whatever numbers we talk about are on the inside. Now, what could cause a charge difference? Ions. Ions carry charges, positive or negative. Positive ions are cations, like sodium, potassium, and calcium. Negative ions are anions, like chloride. These ions are distributed differently between the two environments. For example, sodium is higher outside the cell than inside. Potassium is higher inside the cell than outside. The uneven distribution creates a concentration gradient, which is going to contribute to the membrane potential. Ions can diffuse across the membrane, not through the lipid bilayer because they're water-soluble, so they use protein channels. Diffusion is the movement of a solute along their electrochemical gradient. Chemical refers to the concentration gradient, from high to low concentration. The electro part of this refers to the electrical gradient. If we assume one side of the membrane is positive and the other is negative, a positive ion would move from more positivity to lesser positivity, and a negative ion from more negativity to lesser negativity. And that's a little bit about diffusion that you'll need to know. To understand how the charge difference happens, let's pick potassium. Potassium is higher inside the cell than outside. At this point, we're going to make a few assumptions. One is that the membrane is electrically neutral, so that there is no charge difference. The number of positive and negative ions is equal on either side of the membrane. And the second is that it's only permeable to potassium. When the channel is open, potassium diffuses along its concentration gradient from high to low concentration. So the ions leave the cell. These are positive ions leaving. The negative ions are stuck inside because we're assuming that the membrane is only permeable to potassium. So the inside is becoming more negative compared to the outside. This potential difference that was created by the diffusion of an ion along its concentration gradient is called a diffusion potential. And now there's an electrical gradient in addition to the concentration gradient. Potassium is a positive ion, so the ions would move from more positivity to lesser positivity along the electrical gradient. The direction is opposite, but at some point these two will balance and cancel each other out. The ion stops diffusing and is at electrochemical equilibrium. The potential at which this happens is the equilibrium potential, also called the Nernst potential. This value is different for different ions, but since we're talking about potassium here, this is the equilibrium potential of potassium. These equilibrium potentials are in millivolts and would depend upon the concentration gradients of the ions, outside versus inside the cell. If the membrane was only permeable to potassium like this, then the membrane potential would be equal to the equilibrium potential of potassium. But the membrane is permeable to more than just potassium, and we'll come back to this. The resting membrane potential is a state of rest, obviously. Excitable cells, like nerve and muscle cells, they communicate using action potentials. The action potentials are brief changes in the membrane potential. But in between the action potentials, the membrane is at rest. So the potential at this point will be the resting membrane potential, and it's different in different cells. If potassium were the only permeable ion like how we saw earlier, then the membrane potential would be equal to the equilibrium potential of potassium. But at rest, the membrane is permeable to more than just one ion. So let's add in another, sodium. If the membrane were permeable to just sodium and not potassium, then the membrane potential would have been equal to the equilibrium potential of sodium. If both ions can move, then they're both going to try to bring that membrane potential close to their equilibrium potential, and it'll land up somewhere in between the two. That's where another factor comes in: permeability. The more permeable the membrane is to an ion, then the membrane potential is going to get closer to that ion's equilibrium potential. It makes sense, because if more potassium ions can leave the cell compared to sodium entering, they've got more influence on what happens to the membrane potential. The same thing happens if we include multiple ions. Whichever ions have the higher permeability will have a greater influence on the membrane potential. So multiple ions make their contribution, making the resting membrane potential negative. The value is different for different cells, but for most neurons, it's around negative 70 millivolts. Why is the inside of a nerve fiber membrane negative? That's what we're going to see next. It mainly involves sodium and potassium. On the membrane, there are leak channels for sodium and potassium. Leak channels allow ions to leak through. They are diffusion channels, so it'll be along the gradient. Sodium is higher outside, potassium is higher inside. So sodium enters the cell and potassium leaves the cell. The membrane has more open potassium channels than sodium, so it's much more permeable to potassium at rest. Inside the cell, there are organic anions, like proteins and phosphates, which are negatively charged. They are stuck inside the cell. More positive ions are leaving the cell than are entering, making the inside more negative. So the membrane, it has a higher permeability to potassium, and that's why the resting membrane potential of a neuron is closer to the equilibrium potential of potassium than sodium. I'm going to put down numbers to explain it. So potassium has an equilibrium potential of around negative 85 or negative 90 millivolts. Sodium would be around positive 60. The resting membrane potential of a neuron is around negative 70, so you can see it's closer to potassium than it is to sodium. But this is diffusion, so if the gradients are left like this, then the ions would keep leaking and the gradient wouldn't remain. There's also a sodium-potassium ATPase pump. It's an active transport pump. This is against the gradient. For every molecule of ATP that gets hydrolyzed, 3 sodium ions leave the cell and 2 potassium ions enter. Again, there's a difference. More positive ions are leaving than entering, so it contributes to the negativity as well, but more importantly, it maintains that gradient, and so the negativity. Now, I've magnified it like this just to explain it, but the charge separation is actually happening just next to the membrane. It takes a very small amount of ions to create a potential difference. So overall, the intracellular and the extracellular compartments, they stay electrically neutral. This is just happening at the membrane. Now, this charge separation makes the membrane at rest polarized. In excitable cells like nerve and muscle cells, this potential can change, creating either graded potentials or action potentials. And that is the resting membrane potential. If this video helped you, give it a thumbs up and subscribe to my channel. Thanks for watching, and I'll see you in the next one.